I'm one of the co-founders of E11 Bio. I trained as a physicist, and then something like 10 years ago, I came down to the Bay Area and worked for this guy at his DNA sequencing startup, and that sort of like got me interested in the world of biology. So as a physicist, I'm like very interested in building tools to like measure reality. And one of the coolest things that you can measure in the universe is the brain. It's one of the most complicated objects we know about. So I ended up going down to MIT to do my PhD with Ed Boyden. And as we were approaching the end of my PhD, I started getting very interested in some ideas around how to organize neuroscience work in a particular sort of slice of tool building and measurement and inquiry. So I'm going to talk about that today. So I'm also going to talk a little bit about meta science around like how E11 is organizing work. So to kick things off, let me tell you about E11 Bio. E11 Bio is a nonprofit research organization that is focused on building tools for scalable brain circuit mapping. And we're dedicated to putting those tools into every neuroscientist toolbox. In this talk, I'm going to talk a lot about brain circuits. Here I'm referring to brain circuits as synaptic level connections between cells. This is distinct from, the sort of coarser fMRI measurements between brain areas. So let's just make sure that's not in scope. And this is an important distinction because a lot of the function of the brain is thought to be basically embedded in like precise cell type specific machinery that connects individual cells and gives rise to function. That work? Okay, cool. Okay, so why would we want to map brains in the first place? Well, one reason, and I'll talk about three, is human health. So, there are like 1 billion people in the world, 1 in 8 people is living with a neuropsychiatric disorder of some kind. And yet, when we look at the brain compared to any other organ system, we have the fewest number of treatments in existence, the fewest number of new treatments in the pipeline. Of the treatments in the pipeline, 19 out of 20 of those will fail. And then of the one that actually makes it through, it's actually not going to be any more effective than its counterpart in the 1950s, although it will be safer. So, drug discovery for the brain is broken, and there are a variety of ways in which it's broken that I don't have time to get into all of them here. But one of the more significant ways that it's broken is that we don't really understand at a mechanistic level the difference between functional and dysfunctional circuits. But we are pretty sure that there are circuit disorders which underlie all of these conditions. So, that's something that brain mapping could help. Another reason we might want to map brain circuits is just to do better science. So, many neuroscientists are interested in what is the circuit basis, the substrate of cognition. And these circuits in mammals, they span pretty large distances, millimeters to centimeters across brain areas. But unfortunately, the tools that we have today, the methods that we have, are not able to map these large brain circuits. And in order to scale them up, you would need very expensive equipment, which is out of reach of individual labs. And also, the sample processing approach is very brittle. So if you make a mistake, you have to throw out the whole experiment. So, improved brain mapping would allow neuroscientists, individual neuroscientists, to go and study complete circuits in mammalian brains, and that would unlock new experimental paradigms. A third and final reason why we might be interested in mapping the brain is that, well, it's a bit more speculative, but there is an existence proof in the form of you or me that there are intelligent systems that are pretty low power. I'm running on like 20 watts right now. Pretty data efficient. I can generalize typically from just like 1 or 2 examples, and are generally aligned with human values kind of by definition. By studying the brain and understanding what are the circuit architectures that underlie those properties of intelligent systems like you or me, we might be able to then use that learning in order to build artificial intelligence systems, which are similarly data efficient, energy efficient, and perhaps more aligned with human values. But that's more speculative. Okay, so these are and if people want to jump into questions at any point, feel free. We'll also do Q&A at the end. So these are like big problems facing the entire field, and it's going to take the collective effort of like thousands of neuroscientists to make headway on any of these problems. But, new technology is really interesting in the sense that if it is, useful enough and accessible enough, all of those neuroscientists can take advantage of it. We saw this like in the last couple of decades with DNA sequencing, where now there is no biological laboratory that doesn't have DNA sequencing. So we want better technology in order to let neuroscientists do better science and clinical neuroscientists to make better medicines. And at E11 Bio, we've identified a technology that we think, sort of fits this mold, that has catalytic potential that could affect all of neuroscience. And so, our contribution to the neuroscience toolbox is to make single cell brain circuit mapping in a scalable way, routine, so scalable to mammalian brains, routine and accessible for all neuroscientists. Okay, so what are some characteristics of a broadly useful single cell circuit mapping technology? In my opinion, there are two parts. The first part involves measuring synapse level connectivity in mammalian brains across single cells. So this sort of tells you how the cells are wired up and gives you some insight into sort of the precise connectivity between different cells. The second part is going in and looking inside those cells to map out the biomolecules that give rise to function. So, this tells you things like cell type or cell state or synapse type. It also lets you look for drug targets because, you need a molecule that you're targeting before you can make a drug for it. And so, in essence, what we need is a technology that looks at a brain and gives us a wiring diagram and also gives us the functional components that are wired up by that diagram. So technically speaking, how would you do that? So how would you map brain circuits and biomolecules together in a scalable way? So here's our take on the problem from E11, which involves taking ideas from genetic engineering and from advanced microscopy techniques. So first, we modify the brain itself. We use tools from genetic engineering, like viruses, to deliver different combinations of genes to different cells, which makes them glow different colors, sort of like so. Sort of like so. Is this not working? The animation worked, hooray. Okay, next we engineer the sample, so a tissue sample from a brain, by embedding it in this swellable polymer material. So this polymer can expand the tissue in a very uniform way, isotropically. If you ever played with those dinosaurs when you were a kid that grow up in the bathtub, that's the idea here. It's a similar idea. And so the benefit of making tissue bigger is that things that are bigger are typically easier to see. So we can use much cheaper and much faster microscopes in order to look at these samples. And then finally, we use laboratory automation and those faster, cheaper microscopes in parallel to scan the brain, taking lots and lots of pictures and then stitching them together in 3 dimensions. So and once we have this sort of 3D map, we can use the color codes that we introduced at the beginning to map out the connectivity, kind of like you would map out connectivity on a ribbon cable. So, each neuron has a unique 3D color, and you can sort of trace it, or you can even skip the intervening distance and just look at, the beginning points and the end points. So we also capture the molecular information using a similar color coding approach. So, what's the challenge here for E11? Well, each of these technologies has been demonstrated on an individual basis, and they've been on the cover of Nature and Science and so on. But, they haven't yet been integrated into, a technology platform that, can do all of them together routinely, robustly, efficiently, accessibly. And so, that's out of scope for academic work, which is pursuing novelty. And it's also out of scope for a startup company, because this is pre-commercial. We're not trying to make money off of it. We're trying to give this to scientists. And so that's where the meta science around E11 Bio comes in. So how are we organizing the work that we're doing at E11? In some sense, we're pioneering a model for a kind of nonprofit startup. So, like nonprofit or academic laboratories, we're pursuing open science for public benefit using philanthropic dollars or government grants. And like startups, we have, and unlike academic labs and like startups, we have an unusually high degree of coordination between our teams. So, we have team science and we have, a multi-year road mapping and we have close coordination. So the result is that we have been able to put together sort of like a mission-driven team of very collaborative researchers who are here because they want to see brain mapping made scalable and accessible. And so, a couple other properties are interesting around this. To prevent mission creep, we operate in a very narrow 5-year period of performance. This is borrowed from DARPA, where we're doing neuroscience, we're not selling to the military, but the idea is you want a defined period of performance where you can go as fast as you can, push the ball as far down the field as possible, and then transition it out into the wider world. So on a more meta note, I want to suggest that E11 Bio is just like one of many projects of a similar size and shape. So, there's now another FRO, focused research organization, organized along the same lines that's operating in Boston, and it's focused on synthetic biology. And we have others currently, in the pipeline that will, deploy maybe sometime next year. And so, if you're a researcher and, you're working in academia, but your problem requires too much teamwork, or you're in industry and you would really be prefer to be working on public goods, but you don't want to take academic salaries, the FRO might be sort of like an organizational model that is a good fit for your work. There are a couple of other properties of FROs. So research we've talked about a bit already. In terms of keeping the team focused, it really helps to have, this 5-year period of performance and sort of this North Star for us. It's, making brain mapping accessible. And another thing that's very important is, having a roadmap and a plan for how do you transition the technology out of the laboratory and into the world through different deployment avenues. So, one way to deploy a technology is to, have a startup license the technology and then make a product that every neuroscientist can buy. That's one way to get it into every neuroscientist toolbox. That's what happened with DNA sequencing. Okay, well, I'm going to wrap up then. So thanks, everyone. This wouldn't be possible without the team at E11 Bio, particularly, my co-founder, Todd Huffman, has worked in lab automation for many years and had founded and sold a company before that and is co-leading the effort. And we have great talent from Hopkins and DARPA and MIT and Genelia and so on. So, it's a fun team. We're hiring, we're looking for scientists. You can learn more about us at e11.bio. So thank you.
So tell me, does anyone here have questions? Anyone looking for a job? Oh, Robin.
Ready to talk. You're recording. What's the spatial scale you're looking at recording? You said single cell, but that didn't indicate how many cells at once.
So there are two things. One is, we have these color codes, and so you need to be able to see them. So you need a light microscope. But if you have a light microscope, then you are in this diffraction limited regime where you can't see anything smaller than 500 nanometers or so. So if you expand by a factor of 5 or 6, that gets you down into the sub 100 nanometer regime where you can actually make out these synapses. And when you can make out these synapses,
I didn't mean the scale of resolution. I meant the size of the image.
Yes.
How big is an image at one time?
How many microscopes do you want to buy? So the question, the thing that we're trying to do is, given current estimates, you could probably spend like, $500 million to buy a bunch of electron microscopes and do like 3 or 4 mouse brains per year. We're trying to do at least 10 to 100 fold better than that. So, 500 cubic millimeters per mouse brain times, 100 mouse brains is the kind of scale that we want to achieve. Does that make sense?
So I guess the first part on how
What is the variability that you see from brain to brain? I think this is one of the things that's appreciated from neuroscientists, not so much outside, is just how variable brains are.
Right. So we have to get the data. Right now we have one sort of cubic millimeter of one mouse brain that we've done any sort of connectomic analysis on. But there's a big question about what behaviors are hard coded and what behaviors are learned. And the only way to do that is to be able to scalably map many of these brains and then look at the data and find out. So we don't know.
Yes. But I think that
Can you look at neural circuits in living brains, or do they have to be expanded and dead?
No, they have to be different. So this is definitely like a technique that works on fixed tissue.
Are you looking at techniques that might come up that you might invent for living brain?
I think that's a really interesting area. The physics around how you get information in and out of a living brain is really hard. So I'm not aware of any technology that would actually let us go in and look at it with the same resolution that currently neuroscientists need.
I was just going to say, in response to your question, like living slices might be the way to do it. You can keep brain tissue alive for quite a while. My question is, I think I heard you say the word functional. Did you say that word?
I did say that word. So like biomolecules are like proteins are what gives rise to function in biological systems.
Right. So I guess my question is, how are you defining what a complete circuit is?
What?
How are you defining what a complete circuit is?
Well, I mean, so one question is like, if you can map out a circuit associated with a particular behavior, and for instance, like, given a behavior, you can like maybe take a video of the neurons firing. We have techniques to do that now. And then once you know all of the neurons that participated in that behavior, you can go in with our brain cam technique and take a look at, the structure behind all of those neurons. And so then you can basically just like superimpose all of the neurons that were active in that behavior with the circuit diagram and start to try to understand what is the functional basis for it.
You define your circuit.
Yeah.
I wonder if you could give a summary of what's currently known or hypothesized for how memories relate to physical structure, how thought relates to physical structure, compared to, let's say, a movement of me scratching my head, how that relates to physical structure.
Yeah, I mean, that's a great question. So, we are pretty sure that memories are physically encoded in connections between nerve cells. Like, you can remember the street that you grew up on, and it's been, many years since that. So, that has to be somehow preserved in some structural mechanism over time.
And if I'm recording that, you'd see that circuit firing again and again.
If you had good enough recording technology, you probably would, if you continue to recall that street that you grew up on. But the precise mechanisms by which that memory is encoded is still like a very open question. So this is another case where we need to get the data to the neuroscientists, and then they can start puzzling it over. But before they have the data, it's kind of just philosophical.
And the other ones, that memory, but
what was the other one?
the other one was
Well, I think that's I mean, that's harder, but it's still, probably, I mean, there is still some circuit basis for why you think, because, if you turn off a bunch of neurons, then you stop being able to think. But that's also an open question, and so we need to get the data for that too.
And in terms of consciousness, one metaphor that I've heard is like the brain is a supercomputer, and consciousness is like a flashlight that you're shining on it.
To be entirely honest, I am not qualified to comment on consciousness. That's the hardest problem.
Sorry, I'm just going to squeak back. Hand up for a little while. Sorry, guys.
Thank you for this. This is very exciting. I have been keeping an eye on something called pyramidal cells that in the mammalian brain that have up to 30,000 synapses, and they seem to be perpendicular to the cortical surface and so on. And I'm just wondering how, what hope we have of tracking such a complex cell, if any.
This is something I'm much more optimistic about. So pyramidal cells, like they're defined by they're defined by their morphology, so they're shaped like pyramids. And yes, you said each one has 30,000 sort of outputs, but the technology is the right resolution to be able to identify all of those connections from every pyramidal cell. So the challenge is, how do you scale it in order to actually do the large enough volume to, because these circuits, they span centimeters to millimeters to centimeters, and so you have to do a large volume in order to get to the terminals of those nerves. But that's what we're trying to enable at E11 bio. So very on point.
This question might be obvious for a neuroscientist, but I'm curious, given that the efficiency that you're working on is dead, when you're saying we are isolating a single behavior and identifying neurons that are forming towards it, what's the meaning of the behavior here? Like, how does this compare to, an fMRI measure of being able to understand, tracing through a memory?
Yeah, no, good question. So, MRI measurements are very coarse. So they sort of tell you like which brain areas are firing. But, we're very interested in like, what is the cellular basis? Like, what are the pyramidal cells doing, for instance, for behaviors? But this is where like being able to, integrate multiple technologies and really a focus on technology is what actually gets you to be able to do that. So, there are activity technologies where you get to record from the neuron. So while the animal is alive, you get to see, which neurons are firing. And then, you can operate on fixed tissue later, but you can register those two data sets to each other. So once you get the fixed tissue data set, which is the structural measurement, and you previously got this activity movie, which is the functional measurement, you put them together, then you get structure and function, and you can derive some understanding.
All right, and last question.
Sorry.
So this seems like the absolutely maximally central tendency of research that would be considered to be legitimate by elite academia and by the government and by most government. This seems like the exact maximally central tendency instance of the category research that would be regarded as promising and mainstream, and like where it should go by both mainstream academia and the ultimate philanthropic world and the government, etc. In light of that, like, why would you consider a for-profit or a new organization rather than using more existing? Like, this is exactly what conservative research channels, like not super conservative, like DARPA, are for, or MIT. So like, why be here instead of like at MIT?
Well, the first order answer is I was at MIT trying to do this and it didn't work. So, there's got to be some reason for that. I think a larger question is like, in some sense, it looks and maybe feels like the kind of research that normally gets funded. But in practice, this is actually really hard to do using any of the existing organizational models, because, if you have academics, they only their first authored papers. And so it's really hard to get them to work together as a team. And, government research typically funds work that's basically already done. Like, it's a reimbursement mechanism for research that's already done. So this is, as like a 5-year project, too ambitious to be funded by people grant. So, there are like subtle reasons why this is hard. And one of the nice things about making a new organizational structure is you can just like focus on this particular set of problems that are about like, like teams of like 10 to 15 people working on a scientific problem, and focus on just that. So I want to emphasize, we are a nonprofit research organization. We are not a for-profit. So this is for public benefit, but it's also, we haven't been able to do it in other organizations.
Amazing. Thank you so much. Thank you. Dana. Woohoo.